Good morning, everyone. Thank you for joining us for the second International BioHackathon webinar and the second webinar also. So I'm going to present, I will be the first one to present today. Hope you can see my slides. Yes, you can definitely see it. Great. So I'm Alessandrì Luca, and I'm a postdoctoral researcher here in the University of Turin. And before start talking about the transcriptomics, the count metrics generation, I would like to talk very shortly about reproducibility. Because you have to know, every time you use a code, you kind of think that this code, it will be reproducible on every computer, but that's not the case. So I will just spend a couple of minutes only to introduce you to a problem that I first-hand encountered. So you can be aware that this problem is actually huge. So I was in Boston for a couple of years, and when I was there, the PI there asked me, they told me, hey, Luca, we want to actually do another experiment from, we already performed this experiment in 2011. We want to do that again on other data. The bioinformatics pipeline is pretty heavy, but no worries because we already have all the code. So they gave me the code, and actually the code were on a MacBook Pro of 2011, and this was called a server. So I actually entered in this MacBook Pro, and I found the code. Actually, it took, I had a server in Turin that it will be called Luca's server. Instead, the server in Boston, the MacBook Pro is the red one. I already have a server in Turin that is way faster than the MacBook Pro of 2011. So I was like, okay, I will just transfer the code on the server in Turin and try to reproduce exactly the same results that they obtained in 2011. The main problem was that it took me almost 1 year just to have a running pipeline, and I didn't touch the code at all. The only thing that I did over this year was trying to reproduce exactly the same environment, because I was always encountering problems like this program is missing, this version is not correct, and so on. After almost 1 year, I had the pipeline running successfully. So successfully means without any error. But when I compared the results with the one that they already had published in this 2011, well, you can definitely see that the difference is huge. So basically, these are the common rows. These are the green, that is my server in Turin, and the red one is the server in Boston, the MacBook Pro. And you can definitely see that the common one are way low. And we lose a lot of translocation because, in this paper, we are analyzing translocation. So basically, for each of this sample, we are actually counting how many translocations we have. This is a problem. Slightly differences is acceptable between different versions and also different architecture system. But something like this, this is really heavy. So the main problem, after another couple of months, I found out that the main player in this difference is BLAST. So BLAST is an algorithm that you usually use in order to kind of align, let's say. There is actually a term that is blast, used as a verb. I blast this thing. So you use this thing in order to take a piece of DNA or whatever, and you blast it and check whether this piece align on the DNA, to which organism it belongs, and so on. So it's a very common tool. Also, biologists use it, not only bioinformaticians, because it has not only the API, but also a nice graphical interface online. And the main problem is that, not even the major version, but just the minor one here, it changed. And as you can definitely see, I was using a new version instead of the latest one. And even though I didn't have any error in the code, I had error in the I had error in the results. So why is that? Well, because if you analyze everything, basically from these two versions, it changed not the algorithm, not really the way in which they evaluate the p-value, the e-value, what they call it, but it's a normalization changes. So basically, they changed the normalization from float to int. So of course, if you have a threshold to float, I mean, it will be way different if this is going to be different, like is transformed into int. So algorithm did not change, also the way they evaluate the e-value, but changes the scale basically. And something like this, of course, can mess up everything in the code. So which one is the solution to avoid all of this, to incorporate all this code inside a Docker? So I'm talking about this because, I mean, it's important, and all the code that you will find from this webinar, they're actually on our GitHub from Young Info Life, and they are encapsulated in a Docker. So a Docker is kind of, you can imagine it as, even though it's not a virtual machine, I would say it's a kind, like you can think of it as a virtual machine. So whenever you run a container, a Docker, you are actually running a computer in your computer, kind of. We organized all the code from this webinar in a way in which you can actually access, like run directly the Docker container. So let's see, you can download everything, like you can actually access the GitHub page from this QR code, or simply checking GitHub, Young Info Life, BioHackathon of BITS 2026. You will find basically two main files, like the build one, but you don't really need the build one because we use this from the GitHub. But basically, they run me the bat file and the sh file. The sh file is for Mac, and the bat file is for Windows. So since now, let's check the code. So since now I am on Linux, I mean, I will run it. And I believe it's already running. Yes, it's already running. So you can see that this is, I of course have already all the results, because I mean, doing the count metrics generation is taking a while, even though the data set is from the Drosophila. So it's pretty small, pretty fast, and you can perform actually this this pipeline. I'm pretty sure you can perform it even on your laptop. By the way, in order to run it, as I said before, you can basically click either on this, like either run it through as a bash script, or double clicking on the bat file here, this one over here. You will see some green lines, you will understand that everything is working, and then basically you can access the Jupyter Lab in this localhost 8888. So again, a couple of minutes just to explain how this works. So this Jupyter Lab, as you can see, it's inside a container. So basically, all of these are not folder from my computer, they are folder from the Docker. Besides for this one, the shared folder. This folder indeed is exactly this folder over here. Indeed, if I add a folder or I remove a folder, you will change. Like this folder is basically the connection between the container and your computer. Just be aware that this, sorry, let me close this one, otherwise I will always, okay. So this folder, be aware that this folder, like the folder that is the connection between the Docker and your computer, is going to be exactly the same one in which you have the script. So the script automatically understands in which folder it's in it, and it will share this folder with the Docker. So what do you have here? Basically, you have, okay, you already saw that you can navigate through your folder between this one. And here in the middle, you can change actually the theme. If you don't like this dark, you can take light, like contrast, whatever. And mainly we will use notebook. So you will see that you have the notebook for Python, for R, and also for a specific version of R. You might need this one if you want to run all the code from the second part, okay? Because this very first part over here, we will not really use notebook because we will not use Python, R, or R, but you will use mainly bash. All the script are already inside the Docker, or the script that we use, and are in the notebook folder. So you have part one that it's for my part, and part two that you have all the script for the second part, so the downstream analysis. Okay, let's, and another thing, you can also open the terminal, and if you type bash, you are, you can navigate and do whatever you do in bash in the terminal inside this Docker. And okay, let's start to review some code. And so what do we do, and where do we start? So usually a nice thing for reproducibility is that most of the time when you provide some code, you also want to provide a code to download the data. This is important because, I mean, whenever you make a code, you kind of hard code all the paths, for like where are my FASTQ file or whatever. So if you share also everything, like also the script that you use in order to download the data and organize them in specific directory, well, in this way, everything, it will be 100% reproducible. And this is the only thing that we are doing in this code. So basically, let me increase the size. So basically, we are actually downloading these different data sets that are from, as I said again, from Drosophila. We have some treatment and some control. I mean, I will not talk about this because this will be important for the downstream analysis. So my colleague will gonna tell you more about this thing later on. But yeah, I mean, this script that you can use it like from this case, or you can actually recycle for also other FASTQ to download. Will download this FASTQ. That basically a FASTQ is a raw file that contains all of your reads. Okay, and this one specifically is made for pair-end. So just be aware if you have a pair-end or a single hand. Progress, thread, I mean, I will not discuss all the basically all the options that we can give to all the function, because otherwise this webinar, it will be like 2 hours long, so it'll be pretty fast. But just keep in mind that you have all this code, so you can play with it, and always check the documentation. But I believe that for this webinar, like already having all these things and see how they work is the very best way. Is the very best thing. So the first thing, of course, as I said, is to download this data. And after you run this code, and of course, since it's a bash script, in order to run it, we have to use the terminal. So access the terminal. I mean, you can still use the terminal in this way, but I don't really like it because you don't have color, and so just use bash. And yeah, you can see I'm already in the notebook part 1. As here are all the notebooks, and if you want to run it, simply /1 and then so basically this thing don't have the permission to be run, so you have to chmod, you have to add it the permission to be run. And then if you type download data, I will not run it because otherwise it will override everything, all the data that are already downloaded. It will start. It will take some time, like with a good internet connection, I would say it will take around between 1 hour and 2 hours to download this data. Okay, and this data, there will be in the shared folder and in the FASTQ here. So you will find all this FASTQ parended over here. So the first thing that we want to do after we download the data, indeed, let's check for the second script, because the very first one is another book over here. And also we have to download the reference. This will also download the reference and organize it for you in the shared folder. This is important because, I mean, as I said, this is Drosophila. You have all these FASTQ that basically are. Maybe we can also open a FASTQ very shortly in order to show you how it is, if it's already structured, because I don't remember. FASTQ, yeah, they are still gzipped. It's okay. I mean, the FASTQ, it's basically a file in which you have for each read the sequence and the quality. So basically, each of these sequence of all of your reads has to be aligned to a genome. And of course, you have to download the genome and create what is called an index. And this is basically a second thing that is really mandatory that you have to create this index. Basically, this index over here, what it's doing is downloading. We are not actually creating the index here, we're just downloading the file, but just to tell you the files that you're downloading here, you have one that is the genome and the other one is a GTF. So basically the GTF is a very important file that is required for annotation. Annotation means that for every part of the genome, you have, how to say, you know, actually which in this portion of the genome, for example, there is this gene. In this other portion, there is this other gene, and so on. It's a very big file, and there are actually pretty nice libraries from R, packages from R that can read and open the GTF for you, because otherwise it is a bit overwhelming to open the GTF file for yourself, like if you never opened one before. Okay, so download the reference and the data. And then the third thing, of course, is the sanity check. So you can actually check if your genome, the annotation file, if they are downloaded correctly, how many chromosomes you have, and so on. So, I mean, you can basically run blindly, or these three, they're not really super problematic. Actually, we will start, like the very first important step is the index generation. And basically, I will talk about this parameter over here. So, because this one it's important for the overhang value, and this one usually is the ring length minus 1. So, you know how long are your reads. So, in order to generate the index, this is also important. And I mean, in this case, we have old style reads, so 37 long, so they will be like, this value will be 36. But I mean, most of the time nowadays you have 100, like the read length will be 101 or 76, so it will be 75 and 100. And basically the command is only this one over here, and again, it will use the GTF, it will use the FASTA that we downloaded, and this one we will say to generate the genome. And basically this function is going to create an index. So an index is a way that the computer used to access the FASTA file, and the FASTA is like basically the entire genome, in a more accessible way for the computer, so in a faster way. After you run all of this, so you will see that you will have in the shared folder start index. I mean, it's not really something that you can open it, but you will see a lot of other file created and like this genome basically. And all of these are super important for our algorithm. But I mean, you cannot really check, like open one of that and use it. It's really for the star program that it is required to align. And then let's go to the next step instead. That is pretty important, and this is the FastQC. So before to align, so before to see something and see our reads, where do they belong, and this is basically one of the most important steps from this very first part. You want to see the quality control, you want to perform a quality control of this one. So this FastQC, basically, you can see that there are two scripts, 5 and 5b, because basically you perform the FastQC on every of your FastQ, and then you perform a multi-QC that basically is taking all the results for every of your sample and put them together in a more accessible way. One thing you have to be aware is that since this notebook is not really on your computer, is in the Docker, you cannot open the FastQC row. Here it is. Let's open the multi-QC. FastQC, basically. Yeah, you cannot really open it from here. I mean, because you can still have some values, but you will not have the images. So in order to open it, you have to use basically your computer. So multi-QC, and you can open the report over here. So I will go pretty fast on a couple of things, but I will focus more on some stuff that are really important, but actually I already have them in the presentation. So let's actually use the presentation for this one. So This one is a general statistic that will say the duplicates, GC content, how long are your reads, and as you can see, I mean, not all the reads are the same length, and this is because, I mean, we download several data set, and yeah, this one will tell you actually the unique reads and the duplicates. When we talk about the duplicate, we are talking about the PCR duplicates, sorry, and I mean, we have a lot of duplicates. It's fine. With RNA-seq bulk, this happens. It's not something that you have to be like super worried about, but it's important. All this thing that you will see here, it's something that it's not to take action in some way, but it's to take notes, okay? So it's important. And also this plot is another thing that is super important from the FastQC, because we'll tell you, according to the base pair, so going forward with the base pair, the Phred score, and it is basically a quality of that read. And this is normal that the reads, they just go from a very high to a very low one, even though, I mean, you can see that there are some two sample in particular, that has a very bad quality. Also in the here in the multi-QC report, this is actually very interactive, so you can see which is the sample that has this problem, okay? And there are these two samples over here. Again, it's something that you take notes, so you don't really exclude a priori these two samples. You just take note that these two samples have this problem over here. And again, this one instead of being for position is basically for per reads. So from this one, you can see the overall quality of every reads and per sample, and you can see that really these two samples have some problem. But we will see later on why we don't act now and don't remove these two samples, okay? This one is also another thing that is super cool. Basically, it's the pair-based sequence content. So we know that this one cannot be always like a shade. I mean, in an ideal world, you have a shade color everywhere because you have the position for every of your reads. And then if this is blue, means that for all of your sample, the C was the main value, the main nucleotide, sorry, in the very first position. So we have a trend kind of, but this might be because of the primer used, because I mean, this is whenever we sequence, it's not really already like pure biology. We add something in order to sequence. So the very first base have usually a trend that's normal, but the most important thing is that later on, I mean, we have kind of a shade. That means that the percentage of T, C, G, and A, they are mixed up. That's good, basically. This is also a GC content. This will be plotted against like an expected percentage of GC content. I mean, it's okay. It's not really concerning besides for this one, but it's not like a huge spike, so I wouldn't really, it really doesn't matter. And this is also a pair-based N content. So this N is when not the aligner, but before actually the sequencer was not able to define which base it is. So, I mean, most of the time we have some good percentage besides for this one. We will check later if this will change and why I'm talking about later. Duplication level, again, as before, I will not really go deeper into that. And also this one, it might be important, but I mean, overrepresented sequence, some N means, I mean, this is a sequence that was not a sequence, and then all this A that might be, I mean, for a lot of biological reason, but also there are not a lot of reads, so I wouldn't really worry about this. And then this one is actually really important because it's the adapter content. So this one already check if in your sequence there is an adapter, because actually the following step is going to be to remove those adapter, because if we have to align each of these reads, well, if there is an adapter attached to the sequence, well, that's not going to be like aligned perfectly to the genome, right? Because there is the adapter sequence. So this one is going to tell us if there are like some poly A adapter, Illumina universal adapter, and so on. And this is a status check, like is any heat map. You can see if it's red, yellow, or whatever, or green. Of course, no worries about if there is like, you have to take in consideration this like per type sequence, but mainly I would actually I would be more concerned about one sample, each sample. So I will check this by row. If one sample has a lot of problem, like this one over here, I will not exclude it, but still, I can exclude this like in the downstream analysis, but again. So not all the reads are the same, the length, this we already understood. The overall quality is acceptable. The adapter content suggests that trimming might be necessary, so we have to remove the adapter content. And again, just be aware that all this is required for the downstream analysis. So after you perform your downstream analysis, you have some problem, then you go back to the QC. But so let's go back to the code for the trimming, because what we want to do now is to remove this adapter content. And actually, this is pretty easy because there is a nice tool that is called Trim Galore, trimming, here it is, that perform everything for us. So you don't really have even to put the adapter sequence. I mean, you can put it if you know already what it is. But the cool thing about this Trim Galore is that it will try to auto-detect by itself which one is the adapter, and it will of course trim the adapter. And after you trim the adapter, of course you generate, like, again, all of the files, the temporary files will be generated over here. See, the multi-QC trimmed, it will be generated later, FASTQ trimmed. These are all the FASTQ with also the QC, because we actually say that this one. So it's not only trim, but it will also generate the FASTQC, the quality. And as we don't want to check like each of them, each of the FASTQ, but we want to generate from this FASTQC the multi-QC. So basically, there is the next code, notebook. That is this 6B multi-QC, and this one is basically generating, again, only from that one, the multi-QC report. I'm going to open that, and we will see. Actually, I'm not going to open it because I already have the most important difference over here. So the most important difference is that if you see the overall quality score way increased, so we don't have any more that spike that we had before, and adapter content, nothing. So every adapter is removed, and yeah, so these are the most important thing that you can check. That removing actually removing the adapter actually removed also all the main problem that we had before. Not really going deep again to all other sections, because otherwise it will take too much time. But just so you know that the trimming step is important, it is also important what changed after the trimming step. So now let's go back to the code for the last two steps. And the very first one of the last two is the alignment. So basically, we have to take the trimmed FASTQ, and we have to use a star in order to align them. And all of this, so basically, like you align every single FASTQ, actually not single, like you have to, because they are pair-end, right? So you have to put them together in this script. And then you have to align each pair every time. So that's why there is this for loop, and in which you have like the forward and the reverse one. And indeed, they are also passed through here. And again, I will not go through all of these attributes, but just so you know, I mean, this is a type of how you sort coordinates. This is how you actually filter through the attributes, like what you actually give to the SEM file. Because the SEM, so what is this SEM? And actually, what does this file generate? So this file generates a BAM file or a SEM file. They're basically the same thing, but one is binary and the other one is human readable. So basically are exactly the same thing. And the binary one is smaller, and also for the computer, it's way more accessible to work with binary file. So in this binary file, in this BAM file, you have for each of the reads that were in the FASTQ, it will say, oh, this read aligns to this part of the genome with this quality. And then again, this read aligns to this part of the genome with this quality, and so on. So this part is super important because also the BAM file, if you want to like give it a try, the cool thing of a BAM file is that you can basically take this one and drag and drop on a browser that is called like IGV browser. Is a program that you can download it, and you can see your reads. And sometimes this is super cool so you see where this thing aligned, also how it was aligned, and so on. So it is very important to have this BAM. Some pipeline remove this BAM file after they are transformed in the count metrics, because I mean most of the time you use the count metrics, but I will keep it if you have space. If you don't have space problem. And then let's go to the final one, because as I say, you generate one BAM file for each of your sample, but then this BAM file has to be transformed into a count metrics. And my colleague will show you what is a count metrics. But basically what we want is that for every read that was actually aligned to a gene, it will contribute to that gene. So in the end, we are going to count for each sample how many times we saw a gene. And basically, this is what we do with the very last script. So this very last script, it is a bit different, because it will use, you have to run this matgen sh, and as you can see, it's going to use this R script. So basically, we'll only look at this one. This R script, it might be a bit confusing, only because, I mean, it's going to load all the BAM file, evaluate all the features. So I'm using R in order to do this. There are also better ways to do it, like with Bash. There are some also very old scripts that can perform something like this. The main problem, why did I use R instead of sticking with Bash? Is that sometimes you might have a core dump because of a different architecture. So these things are really, how to say, sometimes they are very old. And some architecture, like the one for the Mac, for example, the ARM architecture, I mean, they are pretty new compared to such tools. So you might still encounter some problem. That's why I moved to R, and I use this feature counts algorithm, because this one, I mean, it will work whatever. And this thing over here is going to generate a couple of things. Like it's going to generate a summary and a count metrics. I'm going to show you only a summary because it's how you read how things are actually aligned. And I will not talk about the count metrics because, again, my colleague will talk about it. So you will find this log final out for every of your samples. So how do you read this thing? The most important that you want to read is the uniquely mapped reads. This is the most important one. So if you have around 80%, I mean, I would say that you are good. Sometimes I also work with less than 80%. And I mean, you work with what you have. So it's not really something that you can do. Of course, you can modify some of the parameters, but most of the time, the parameters that I use in this pipeline are very standard ones. So I wouldn't change unless something really happened. So this is the most important actually value that you have to look into that. Then you have all the mapped reads, multiple mapping reads. So mapped reads are actually because they are maybe too short or they have too many mismatch. I mean, here, not a lot, but a lot are too short. And also there is this multi-mapping reads because one read can be aligned to multiple parts of the genome. So you are actually unsure about where these reads belong. So if it's unsure, it will not be put in the final one. Okay, so for my part, I hope I didn't take really too long. For my part, I'm done. And yeah, please, if you have any question or trouble with downloading the Docker, open an issue on the GitHub or write an email. And I will leave the stage to Davide. Thank you.
Hi everybody. Thanks, Luca, for the first part. I'm Davide Bressan, and I am a postdoctoral researcher at the University of Trento. I'm gonna focus now on the downstream part, so starting from the row count matrix to everything that happens after. So let me load the slide. Here we go. All right. I wanted to first show you how a count matrix looks like. So you can see here on the right that we have the count matrix look like this. So we have on the rows the genes, and then on the columns of the count matrix our samples. And then for every gene we have an integer number that corresponds to the amount of reads that align on that gene. And this is why we call row counts, because these are the fragments that align on a gene without applying any transformation, any normalization yet. Then, along with the row count matrix, we also have a metadata matrix on the right here, where we have now our samples on the rows, and then on the columns we have metadata information like the type of treatment that was carried out on the biological sample, or on a patient. Then we can have clinical variable like the sex, the age, the patient, as well as many other technical variables like the time of collections, the operator, and so on. And so in this part, we're going to start working on the count matrix. We're going to perform normalization of the row counts. We're going to see how to transform the data to make them suitable for then exploratory data analysis, which is basically PCA and clustering. And finally, I will briefly mention how to actually remove confounding factor in our dataset. And so before I will show you some slides, and then in the end I will move to a notebook where we can look also the code of how to do these steps. Sorry, all right. So there are several types of samples that we can encounter while performing analysis of RNA-seq, bulk RNA-seq specifically, but also, of course, single cell. And we can have mostly, they differ based on the perturbation strategy that is applied in the lab. Sometimes we find the sample derived from knockout of a specific gene. But we can also have knockdown of a gene, and the difference is that the knockout happens at the DNA level. So, for instance, with a CRISPR-Cas9 approach, where we completely abrogate, for instance, an exon or the promoters to avoid that the gene can be transcribed. On the opposite, knockdown is when we partially reduce the expression of a gene by using approaches like RNA interference. And here on the right, you can see briefly how this works. So you have a protein complex called RISC that, together with a small interference RNA that is made to recognize a specific mRNA for a gene, will bind this mRNA, and then the mRNA of interest will be cleaved, so it will be basically cut in a specific point. And when it's cut, then it's fragmented, and this will lead to the degradation of this mRNA. And so in general, a knockout will probably induce a stronger change in transcription, in the transcriptional level, because it's a stronger effect compared to a knockdown. But yeah, this cannot always be the case. And then it's really important to know the temporal resolution of our sample, meaning that if they were taken at a steady state, or if they're coming from a time series. So steady state means that basically, let's say you have cell lines in culture. You treat one cell line with treatment, and then you have your wild type, and you collect the sample from the cell line at the same time, and you're basically capturing a snapshot of a biological state at a specific time, and that's it. However, you can also have experiments where you actually collect samples from the same cell line at different time points, and then in this way you can then check for changes in expression that are present along a trajectory, basically. And the dataset that I will use for this tutorial is the Pasilla dataset. And the Pasilla is the Drosophila gene that is actually knocked down in this article here, in this work shown on the right. And in this experiment, they collected 7 samples, 4 from untreated, so wild type samples, and 3 from samples treated with RNA interference, so to achieve a knockdown of this gene, which is actually a splicing factor that is involved in the splicing complex, and so to regulate alternative splicing in the cells. And also this data is at steady state, so we don't have temporal resolution. And of course, along the row count matrix for these 7 samples, we also have some metadata information that we will look at more in detail later during the notebook part. Let's start dig into how and why we want to perform data normalization. So basically, we want to normalize the row count because we want to remove the unwanted differences, and unwanted I mean that are not biologically relevant, so they're just due to technical factors like the coming from the sequencer or from the error in the analysis. And so, for instance, one of the first factors that we need to take into account and we need to normalize for is the sequencing depth. You can see here on the right, we have 2 hypothetical samples, sample A and sample B. And as you can see, sample A has overall a higher number of reads aligned on its gene. So this means that probably sample A has a higher sequencing depth, so recovered during the sequencing part, recovered overall more fragments compared to sample B. And of course, if we don't account for these, we could think that a gene is more expressed in sample A than sample B, but that's not the case because it's just due to a technical difference. And then another aspect that we usually want to account for is the RNA composition. And RNA composition is related to the fact that sometimes you have a few highly differentially expressed genes, that, like the one down here, called gene DE, that take up a huge amount of reads. And then if we normalize by the total number of reads in sample A, this could lead to the gene X to show a lower expression compared to sample B, just because the huge amount of reads taken up by these very few genes skew the normalization, basically. So this is another factor that we need to be careful with when we perform the normalization. And finally, in some applications, but not for differential expression analysis, we also want to normalize for gene length, because of course, if a gene is longer, so it has more exon, it could be that you have more reads aligned on it just because it's longer, so it has more exon overall. And how do we achieve the normalization? Well, there are several methods that have been proposed in the past years. And one of the most common one is called the median of ratios, and is within the DSEC2R package, which is one of the most used package for differential expression analysis in R, but it's also now available in Python. Of course, we have other methods. Quite common is also the trim and mean of M values, which is present in the Edge R package. And we also have other normalization like counts per million and transcript per million. Median of ratio and TMM, they account for sequencing depth and RNA composition, while CPM and TPM, they account mostly for sequencing depth, but not for RNA composition. So they are usually not suited for differential expression analysis, but they can be used for visualization and exploratory analysis. Once we normalize our row counts, so we obtain a normalized version of it. Another transformation that we need to apply is important because the counts data have a property that is called heteroscedasticity, where basically you can see in this plot when the counts grow, the variance of the count grows with it. So basically, this property needs to be corrected because several downstream tools assume instead that the data is homoscedastic, and so that the variance is constant across the different range of expression. And so we need to transform the data to be able to achieve a homoscedastic dataset. And to do so, one of the simplest transformation is the logarithmic one, It's quite fast, intuitive. And you can see here now that by applying the transformation, the high value of expression, we do not see high variance anymore. However, the logarithm transformation introduces a higher value of standard deviation in the lowly expressed genes. And so this can also be, so it doesn't completely correct for the heteroscedasticity. And so in the DSEC2 package, another transformation, which is called BST, has been shown to be more effective in correcting the heteroscedasticity. And as you can see here, indeed on the right, also for the low counts value, now the variance it's lower, and this is the most suited transformation that we will also use later in the coding part. I will show you how to actually run it on your data. Then once we transform the data, we can move on to the exploratory data analysis. And one of the first thing we want to run is the principal component analysis. And this is really important because it can help us to pinpoint the presence of outliers in the dataset, and also to check if actually our samples are clustering together, are similar, based on for instance, the biological condition, time point, and so on, any variable metadata variable that you can think of. Basically, the PCA is a dimensionality reduction technique that transform the 1,000 of variables, so your genes that show higher correlation into new variable, which are called principal components, that capture the maximum variance in the dataset. And then for each principal component, here on the right, we are plotting in a scatter plot the PC1 versus the PC2. Each of them explain a part of the variance. In this case, here you can see the PC1 explain around 60% of the variance, which is quite a lot, followed by the PC2, which instead explain around 30% of the variance. And if we color the points, which are our samples, by the biological conditions, we see that PC1 explain mostly the differences due to the biological treatment, so the RNA interference, while on the y-axis, the separation that we see, so along the PC2, are driven by the type of sequencing that, in this case, is either pair-end or single-end, which I will explain in a second what it means. So indeed, PCA is really helpful because tells us if we see, for instance, a sample that is completely far away from the others, that could be an outlier that we might need to consider, to exclude, or to handle in the downstream analysis. Then another check alongside the PCA that is quite common is computing the distance between our samples. And one of the most used one is the Euclidean distance that we apply on the variance stabilized transform data. And this can quantify how similar are, in terms of gene expression, of course, our samples. And then once we compute this pairwise measure between all pair of samples, we can visualize them on a heat map here on the right. And here in this heat map, if the color is darker, it means the two samples are more similar because their distance is smaller. And then if we apply hierarchical clustering on this heat map, we see that again our sample cluster first by biological conditions, so in red and blue here, followed by the type of sequencing, so our technical variable. All right, so again this check is important to check for the presence of outliers, or if we see, for instance, that we expect some samples to cluster together because they were treated with the same compound or with the same drug, and we do not see good clustering between them, could be an issue in the experimental part that we need to discuss, we need to address for. And then finally, for this introductory slide, the PCA also can help us, as I was saying, to pinpoint batch effects. So for instance, the difference that we see in the type due to the type of sequencing could be considered a batch effect. So something that drives difference in the data set but it's not due to the biological condition, but just by a technical effect, which could be, for instance, the date when the experiment was carried out, who carried out the experiment, and so on. So there are many batch effects that could show up. So one of the most used way to correct for batch effect is the SVA package in R. And indeed, if we observe a known batch effect, so we see separation by, for instance, the type of sequencing like we saw before, we can run combat-seq is a function in the SVA package that adjusts for this batch effect by while preserving the real biological difference in the dataset. However, if we do not know what is driving the separation between our sample and the PCA, we cannot find any metadata variable that is actually driving the batch effect. Again, we can use another function in the SVA package, which is called SVA, which can help to identify hidden and technical noise that we don't know where it's coming from, and correct for these to avoid to have then an impact on the biological, on the downstream analysis again. All right, so now let's move to the R Markdown notebook, where I'm going to show you the code. Again, as Luca was mentioning before, you can find the notebook and also the package and with R already installed within the Docker container that is available on the GitHub page over here. Okay, so let me now share the notebook. All right, so here we go. You should be able to see. Yeah, you should be able to see the notebook. All right. So let's start by, of course, the packages, the R packages that are needed for the normalization and the visualization of the dataset. For this, in this notebook, I used the complex heatmap package for the heatmaps, and then the package dsec2 for the normalization and the transformation of the row count data. And then ggplot2, I also use it for all the graphic and the visualization, basically. Let's start by loading the count matrix into R. So this is basically the output that was generated by the alignment, like Luca showed before. And in this case, we load it within R, and it's stored within a TSV file. And we can load it with the read table function. And this is what it looks like. Here we're only showing the first 4 rows of the dataset, so we have only 4 genes. And then we can see on the columns the 7 samples. And for each of them, we have the quantification of gene expression. We can check the dimension of the row counts table, and we see that we have about 15,000 genes on the rows, and again, the 7 samples on the columns. Then we need to load into R also the metadata table. And in this case, it's a CSV file, and this is how it looks like. We have several columns for each of our sample. We can see the sample that were treated with the RNA interference and the one that were not. Then we have technical variables, so the type of sequencing. In this case is either single end or pair end. So pair end is now more common. It's basically you have 2 mates, so 2 reads for every fragment of mRNA that are aligned on the 2 ends of the fragment, while single end you only have 1 read per fragment. Then we have other metadata variable that we don't look at for now. And as you can see, however, the sample name here, treated1FB, is different from the sample name that we have in the row counts matrix, which is treated1. So, of course, we need to correct for this because we need to match the samples in the row counts table with the samples in the metadata table. So here, down here, this line of codes here are needed to actually match the sample names that is present in the annotation table with the one in the count tables, and then also to order the samples in the annotation table to match the order in the row counts table. So these are just check that we need to do to make sure that we have everything in order before moving on. Once we align the metadata table with the row counts table, we can move on and we perform a filtering of genes. And specifically, we remove genes that show zero or really low expression. And this is recommended for improving also the computational efficiency of the analysis, because if we have many genes with many zeros, they are not informative. And so we can remove them by setting a threshold. For instance, this X2 package recommends to remove the genes that show really low expression, like in this case, we set a threshold of 5 reads in at least n samples, where n could be the size of the smallest group of sample. In our case, it's 3, because we have 3 treated samples. So we are going to remove genes that do not have at least 5 reads in at least 3 samples. And as you can see here, we go from the 15,000 genes to around 9,000 genes. So we remove many rows that are basically non-informative. Once we do this, we move on to the normalization part. And you can see here that we have the total number of reads per sample shown. So for instance, for the sample untreated one, we have about 14 million reads, while for the untreated two, we have about 22 million reads. So the difference is quite huge. And this is exactly the difference in sequencing depth that I was mentioning before. So we are going to use the median of ratio method for the normalization. That is the one implemented in this X2, which basically will calculate the geometric mean of the counts for each gene across all the samples. Then we basically compute a ratio of the gene count in a sample, or for the geometric mean over the geometric mean. And then by taking the median of all this ratio, it computes the size factor that we'll use to normalize the sample. And so this normalization will account for the different sequencing depth and in RNA composition. Let's see how to do it in R. So basically, since we are using DSEC2, we need to create an object of the class DSEC2, and we can do this in this line of code here, where we basically provide count data, the metadata here as call data, and then we provide the variable that the known biological variable. In this case, is conditioned, where we have the information about untreated and treated samples. Then we can run the estimated size factor, which will return and store within the DDS object the normalization factor or size factor, and we can actually print them by calling the size factor function, and they are shown down here. In general, if the value is greater than 1, it means that sample was sequenced deeper than average. And indeed, you can see the sample untreated 2 as a size factor of 1.8, which is quite high compared to the untreated 1. And then to actually retrieve the normalized counts, we use the counts function in R by providing a parameter normalize equal true. And in this way, we now have for our genes, we have the normalized counts, which are not integer anymore, but now they're float. And so in this way, we achieved to normalize our row count matrix. Next, as I said before, in order to perform the downstream analysis, we also need to transform our normalized counts and to perform the variance stabilizing transformation. And we need to do this again to correct for the heteroscedasticity of that is intrinsic in the count data. And we can check by ourselves actually this the heteroscedasticity by computing for every gene in the dataset, so our 9,000 gene, the mean and the standard deviation. And then we can plot in a scatter plot with the ggplot function. We can plot the relationship between the mean and the standard deviation. As you can see, as I already mentioned before, growing value of expression also show higher standard deviation, which is something that can impact the PCA and the clustering, as well as network analysis. And So to correct for this, it's just one line of code. We call the VST transformation on the DDS, so on the DSEC2 object. And now on the VST transform counts, basically we went from row counts, normalized counts, and now VST transform counts. We can compute again the mean and the standard deviation and plot again the relationship between the mean and the standard deviation. And we now see that the heteroscedasticity is gone, and we achieve a good and regular standard deviation across all the different layers of expression. Then the exploratory data analysis part, so EDA. It's quite easy to do in R. We can plot the PCA by providing the variable in the metadata that we want to basically group our samples by. And again, by using the ggplot function, we compute the PCA, and we can see for each sample exactly where it lays on the PCA map. Again, we see that the major difference is driven by the biological treatment, followed by the type of sequences, so if it's either pair-end or single-end. Of course, we could also plot the other principal components, PC3, PC4, and so on, but usually since they explain a lower portion of the variance, they are less informative. Then let's look at how to do the sample distance heat map. We call the dist function in R. In this case, we need to transpose our VST transform matrix because the dist acts on the rows, but we have actually genes on the rows, so we transpose, and then we get distance matrix where we have for each pair of samples how much differ they are in terms of gene expression. And then by using the complex heatmap package, we can actually build our heatmap with the clustering. In this case is a complete linkage clustering based on the Euclidean distance. And again, we see that the samples separate mostly by condition followed by type of sequencing. And so in this dataset, even if there is a slight technical deficit due to the type of sequencing, we still see the PC1 and driving the it's due to the biological difference, so this is good. And also we see that we do not have outliers or samples that cluster completely separate from the biological treatment. So confirming that this is a good dataset where we can move on. Then finally, we can also collect which are the genes that show the higher variance in our dataset, and we do this by running the row-vars function in R. And then basically again we can plot an heat map where we have on the row our genes from the Drosophila, and on the columns the condition and the type of sequencing. And in this case, we can see that, for instance, we have some genes that are only that show similar pattern across the untreated and treated samples. For instance, we see the gene 3360 here that is basically down regulated or shows a trend, because here we do not have a statistical value associated with it, so it seems to be lower expressed in the treated compared to the untreated samples. And indeed, this is the gene SESB, which is also mentioned in the original paper from the Pasilla dataset, while the gene ANT2 is actually shows a higher expression in the treated compared to the untreated. But then we can also see that some of the genes are actually impacted by the technical difference. For instance, so the type of sequencing, for instance, the 29856 samples here, we see a quite big difference between the single end and pair end. So we have to keep in mind that this could be a confounding factor for later analysis like network and so on. All right, so this is it on my part. I hope I show you that how we can achieve a good normalization of our row count metrics, how we can transform our data to avoid the heteroscedasticity, or at least reduce it, and how to actually perform exploratory data analysis to see that the experimental conditions that we and the experimental part that is actually correct, and then we see a good clustering among our samples, and we are able to spot outliers. So with this, thank you for your attention, and I wish you good luck for the biohackathon.